This is chapter 5, notes part 2. Remember, we're talking about the large biological molecules. In this one, we're going to be talking about proteins and nucleic acids. Proteins account for more than 50% of the dry mass of most cells. Enzymes are proteins and speed up chemical reactions in cells. Proteins are constructed from monomers called amino acids that are then folded into a specific shape that fits its function. Amino acids are structurally all the same except for the R group, which is one area that is different in all 20 amino acids. You can see page 79, figure 15.6. It shows all the amino acid structures. You do not have to memorize all of the amino acids. Figure 78, or page 78, figure 5.15 outlines some of the jobs of proteins. There are so many proteins in the cell, and they do lots and lots and lots of different things. We're going to spend a good unit, no, not a whole unit, but a part of the unit talking about how our cells build these proteins. How do we take the amino acids and build these large proteins from it? Because proteins are so important in everything we do. For example, enzymes. All enzymes are proteins. They speed up chemical reactions. Without them, our digestion wouldn't happen fast enough for us to survive. We wouldn't be able to build proteins fast enough to survive. So they're important in making sure things happen fast enough for us. Defense. So your immune system is largely made up of proteins. Storage, transport. Think about in the cell membrane, transporting things across the cell membrane. Receptors on the cells so that you can recognize signaling molecules. Hormonal regulation is by proteins. Contractile and motor functions of different cells. Structural. All of these are different things that proteins do. We are made up of largely proteins. This just goes a little bit of an overview of what that function is. So enzyme proteins, the function is to speed up chemical reactions. Defense is protection against disease. So examples would be antibodies that help destroy viruses and bacteria. Storage function, for example, the storage of amino acids. Casein is a milk protein, is a major source of amino acids for baby animals. Plants have storage proteins in their seeds. Ovalbumin is the protein of egg whites used as an amino acid source for the developing embryo. Transport proteins, the function is the transport of substances. Hemoglobin, an iron-containing protein in vertebrate blood, transports oxygen from the lungs to other parts of the body. Other proteins transport molecules across the cell membrane. Hormonal proteins, coordination of an organism's activities. Insulin is a hormone, and it is a protein secreted by the pancreas. Not all hormones are proteins, keep that in mind, but some are. It causes other tissues to take up glucose, thus regulating your blood sugar. Receptor proteins, response of cell to chemical stimuli. Examples, receptors built into the membrane of a nerve cell detect signaling molecules that are released by other nerve cells. We'll talk about that more when we get to the nervous system. Contractile and motor proteins, think about actin and myosin and how they create the contraction of movement. Structural proteins, support is their function. Keratin is a protein in the hair, thorns, feathers, and other skin appendages. Insects and spiders use silk fibers to make their cocoons and webs, respectively. Collagen and elastin proteins create a fibrous network in animal connective tissue. When two amino acids join, they combine with a peptide bond by a dehydration reaction. Remember, dehydration is going to be the synthesis of something. So we're taking small amino acids and creating a larger protein. Dehydration is going to remove a water. That's why we call it dehydration. And they're going to be combining. The peptide bond is going to bond those two amino acids together. Who remembers what a dehydration reaction is? I already just went over that, so I hope you do. Proteins have primary, secondary, tertiary, and sometimes quaternary structure. It is the final 3D shape of the protein that determines its function. Remember, shape, function. Shape determines function. Page 82 and 83, figure 520, will show all 4 levels of structure. I think I have it in here too. The function of the protein depends on its ability to recognize and bind to some other molecule. Many diseases are associated with misfolded proteins, so the protein did not fold correctly, and therefore the protein does not function correctly. How is this important? Here's an example. Sickle cell disease is due to one amino acid change in the primary structure of the protein, which affects the secondary and the tertiary structure and causes red blood cells to form a sickle shape and clog blood vessels. I think I have a picture of that too. So really quickly, these are the amino acids. You don't have to memorize these, but there are lots of them. They're all the same except for their R group, and that's going to be this here. So you'll see that in yellow. The rest of it is all the same. And then here in green, and then here in pink, and then here in blue. And the reason is because they group the amino acids based on some of their properties. This just shows that dehydration reaction, where we're removing a water. And then this bond here that's now going to connect the C and the N after this part was removed is going to be a peptide bond. This shows a ribbon model of a protein in a space-filled model. This shows a primary structure of, so this is like, you can see the actual physical atoms of the amino acid. And then they just get kind of lazy in drawing those, and then just show the amino acid chain. And there can be thousands of amino acids in a protein. And this just shows one there, they're looking at like 127 or so. But there can be a lot of amino acids in a chain. So the secondary structure is when you take that chain and it folds either into an alpha helix or a beta pleated sheet. And then the tertiary structure is when that is going to then again fold and form into a specific shape. And then quaternary doesn't always exist, but this is where you take one and then a different protein, and you actually combine the two pieces, the two polymers. So this is that sickle cell case. Here's your primary structure that is just simply the amino acids, the list, the order of amino acids. And this amino acid, this number 6 here, is different. Instead of having leucine, which is what a normal person's blood cells would have, sickle cell individual would have valine, and that causes the secondary structure to be different, as you can see here. And then that causes the quaternary structure to be different. This is where we have multiple chains coming together. And ultimately, the function is going to be different because it messes up the shape of the red blood cells. And these guys don't carry oxygen very well. And then, well, not no oxygen, but not very well. And therefore, individuals that have sickle cells don't get oxygen to the rest of their bodies like they should. Denaturing a protein can happen when the protein becomes biologically inactive. Interactions with the protein structure are destroyed and the shape is no longer functional. It can result from excessive heat, chemicals, pH levels being out of range, or anything else that is not normally where it should be. Body temperature is a great example, and excessively high fever will cause the multitude of enzymes within the cells of your body to denature, and death can occur. That's why if you get a really, really, really super high fever, like 105 or so, you need to go to the hospital, because it will cause those enzymes that are supposed to be at basically, 98.6 Fahrenheit. It will cause those to denature, and then your body's going to just shut down. So it shows normal protein denaturation. That's going to cause that shape to unwind. It's going to cause that shape basically to fall apart. And remember, shape determines function. So if the shape falls apart, so does the function. Determining the 3D structure of proteins is not easy. Some methods like X-ray crystallography or magnetic resonance spectroscopy have been developed, but there is still a lot of unknown, and the structure of determining these structures may help cure diseases like Alzheimer's and Parkinson's. Folded is a really cool program. If you look it up on the internet and you go to it, it basically is we've uploaded all these protein sequences, and people just play around with them. And hopefully they hit something that actually is relevant to the scientific world. So they actually enlisted basically the general public that just likes to play around with this, and they made it kind of a game to kind of try the different ways that the structure can be put together. And then they look at it and see if that's actually something that can be viable. It's really hard to determine the structure of a protein. It takes a long time. So we don't even know half of the proteins that exist, the structure of them. Nucleic acids. The two nucleic acids are going to be DNA and RNA. They are made up of nucleotides. These are the monomers. DNA and RNA allow organisms to pass information from one generation to the next. The flow of genetic information is as follows. DNA is transcribed into RNA, which is translated into the protein. We will spend a whole chapter on this. Nucleotides consist of 3 parts: the nitrogen-containing base, a 5-carbon sugar, and 1 or more phosphate groups. DNA is double-stranded, RNA is single-stranded. They run anti-parallel. One end is 5' to 3', and the other is opposite. We're going to explain what that means in more depth when we get to our unit on DNA. DNA carries heritable information in the form of genes. The sequence of bases in the genes determines the overall amino acid sequence and thus the protein structure. This is protein synthesis, which we will discuss much later in the year. So here is your DNA. This is only one side of the DNA. 5' actually is where the carbon is at. So we count the carbons in the ring here. And you have a carbon here, 2, 3, 4, and then 5. And because this carbon is actually pointing up, we call it the 5' end, because that particular carbon is hanging out. The 3' end is here because you got 1, 2, 3, and this is your third carbon. Sorry, I made a mistake. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Your 5 end is sticking up there. Your 3 carbon end is sticking down here. Now, the opposite end side of the DNA will run the opposite. This will be your 5', this will be your 3'. If you don't quite understand that right now, don't worry about it because we will talk more about this when we get to DNA structure. So this is your 5' carbon up here, and this is your 3' carbon down here. And then if this carbon is pointing down, it's 3' end. If this carbon is pointing up, it's a 5' end. And then your bases, you probably heard these or remember them from DNA or from Biology 1. A, G, T, and C. And then RNA actually has uracil instead of thymine. Your sugars, DNA comes from the term deoxyribose and ribose from RNA. Those are the types of sugars. So if you look at a nucleotide, which is right here, you've got the base. Okay, that's either going to be A, T, G, or C. You've got your sugar, which is either going to be deoxyribose if it's DNA or ribose if it's RNA. And it's also sometimes called a 5 carbon sugar because it has 5 carbons. 1, 2, 3, 4, and then this fifth one sticking up. And then they have a phosphate group attached. So those are the 3 parts of the nucleotide: the base, the sugar, and the phosphate. And this just shows some different ways of looking at DNA. Notice this is the double helix structure. This gives you more up close showing how the bases are paired, C's and G's and A's and T's. And then this is giving you an example of transfer RNA and how it's shaped a little bit differently. And that's it for this video.